Hello, how's it going today, everyone? Here to record the next lecture. My little dog's here trying to get in the picture as well. So if you hear snoring or farting or barking, you know that's what it is there, yeah? So let me get started with the discussion today. Today, we get into the idea of blood. So a lot of you have the idea that blood is this liquid that flows through the body, and it is, but it's a lot more complex than just this red fluid flowing through our system. So when we attack this, all the chapters, yeah, so we're gonna kind of look at them in a couple of different ways. I'll be very forward right up front and say, I'm not gonna teach this course strictly as a nursing course, right? So I know a lot of you need the material for nursing, but also some students need it for their different programs. So respiratory tech, sonography, physical therapy, coaching, that kind of stuff. So I'm gonna present, it's a general introductory anatomy physiology course. So I'm gonna present it as such. So not a very specific type of nursing program discussion. And by that, I mean, in the nursing realm, it's a lot of sort of ranges, right? We want to keep this within this normal range. If it goes above, we have to medicate. If it goes below, we have to intervene, that kind of stuff. So the book and the notes do present ranges. I will present ranges. There will be some homework questions that sort of necessitate that you know those ranges, but again, I'm not going to focus on the numbers. We're going to focus on the concepts, and hopefully then you understand at least what that range is for. Right? But again, that's the way I'm going to proceed through the semester. So we look at blood, right? Blood is that type of connective tissue that you should have reviewed a little bit or thought a little bit about with the last assignment. So blood is a connective tissue. It doesn't seem like a connective tissue because it's liquid, but it does have the matrix and the formed elements within. So here we're looking at a sort of a combination of plasma at about 55% and about 45% of the formed elements, or the actual cellular or solid parts of the blood. So 45, 55 is an average breakdown of the composition of blood. So within the blood, right, regular English, we say we have red cells, we have white cells, we have platelets. The book's going to get a little bit more detailed. Instead of red cells, we're going to call them erythrocytes. White cells, leukocytes. And we're going to distinguish between several different types of leukocytes. And then these little fragments, cytoplasmic fragments, that we call the platelets, right, or thrombocytes. So physical characteristics of blood. So pH, right? So if you remember pH from 1306, neutrality at about pH 7, blood is not far from there, about pH 7.4. Again, there's our little ranges. You notice we want to try to keep it in the neutral range. A lot of these gimmicks, these fad diets, alkaline water, these different types of foods that you're supposed to eat that's supposed to shift your pH, all that can be actually dangerous, right? We need to be very close to this, this neutrality range there, right? Volume, right? So ladies, you have between 4 and 5 liters of blood. So imagine you go to the store, Albertsons, Walmart, Target, wherever, and you buy those 2-liter or 3-liter bottles of soda. So you can see that's about how much blood volume is circulating in your body, 4 to 5 liters. Males on average tend to be have a larger body size. Again, not always the case, but on average, males between 5 to 6 liters of blood. So, and obviously the larger the person, Shaquille O'Neal probably 8 liters of blood, right? But again, the range, the normal range is about 4 to 5 for females, about 5 to 6 for males. Functions. This is a general overview, right? We're going to look at these throughout the semester, integrated into different chapters, right? So the distribution role, right? Blood will distribute oxygen, carbon dioxide throughout the body. It will distribute nutrients, waste products, waste gases, different hormones from the endocrine system, right? The endocrine system utilizes the blood as that transporting capacity. Regulation of body temperature, the acid-base buffering system of pH regulation, with the help of the kidneys trying to maintain the proper fluid volumes, right? And the leukocytes themselves have a protective role for against infections. The platelets can help with the loss of blood by clotting. So lots of different things that we can look at throughout, the discussion on blood and integrate it into other chapters. Okay, I'm going to use this term often, the hematocrit. The hematocrit is the different separation, right? So let's say the phlebotomist takes out some blood. We have this sample of blood. The blood is centrifuged. So what it does, basically, based on the volume, or I should say the mass of the components. So the iron-containing erythrocytes, or red blood cells, are heavier, so they will spin. As we're spinning, they go farther into the test tube. The plasma stays on top. So in a normal hematocrit, we should end up with 3 separate distinctions, right? What we call the packed cells, the red blood cells, the buffy coat, which are the white cells, and then the liquid component, which is basically the plasma. So plasma, the lightest, leukocytes, and then the iron-containing erythrocytes, which are the heaviest. So again, there's the nursing ranges, but on average, right, you can see that sort of 45% is in between both. So on average, we should expect about 45% of red blood cells in hematocrit, and again, about the 55% of plasma in normal hematocrit, and a very, very small, less than 1% of the buffy coat, the white blood cells there. So plasma, what is plasma, right? Plasma is basically water, right? It's water with a bunch of dissolved components, nutrients, waste, gases, hormones, ions, proteins. So anything and everything that's in your body floating around will be dissolved in the blood plasma. Again, so very separate from the white cells and the red blood cells. So within the plasma, right, so let's look at some of these proteins. These are significant. We have a couple of proteins that we're going to address again in other chapters. So the albumin, the carrier proteins, this will be significant when we cover the respiratory system. Globulins, the basically antibodies, right? We're going to address those specifically in the immune chapter. And we'll get into this a little bit later. Fibrinogen, these are clotting proteins necessary for hemostasis, for the stopping of blood. Right? So they're able to sort of patch up the damaged blood vessels and prevent further bleeding. All right, so let's go back to our formed elements, right? So the 45% of the hematocrit. So of the 45%, 95% of those are the erythrocytes, or the red cells, 5% being the white cells, and a very, very less than 1% fragment of the platelets, right? So we have a normally right stained picture here. These are the red blood cells, and then these are the larger white blood cells, and we're going to have a complete discussion on those in a bit. All right, so as they say, in the beginning, right? So we know that we have blood, but I don't know if you've ever contemplated, how did this blood develop? How did it form? You know, how do we go from a DNA sequence to the actual cell, right? And this is where we have to discuss these basically stem cells, right? And this process, hematopoiesis, sometimes called hemopoiesis, the blood, right? Hemato or hemopoiesis, basically the formative process of. So hematopoiesis, the formation of the blood. And this process will occur in the bone marrow. So that's the where. Where does it happen? It happens in the red bone marrow. So the largest bone in our body, if you remember, the femur, very, very good site for hematopoiesis, as other red bone marrow areas in the body as well. So before we can have blood, we must first start off with this stem cell. The stem cell, which doesn't know what it's going to be when it grows up. It's a very plastic, very flexible, very adaptive type of cell that, depending on what instruction it receives, what gene gets triggered, basically it will then become a variety of different types of cells. So this cell is a stem cell, but it has already committed to becoming a type of blood cell. So it's not, there are more basic stem cells, even more flexible stem cells that can become nerves, that can become muscles, that become different things. This one is already committed to becoming a blood cell, but it doesn't know which blood cell it's going to be at this point. But I do want you to know this. There's going to be several homework questions involving the hemocytoblast. Right? Hemo, blood, cyto, cell, blast. If you remember the difference between blasts and clasts, clasts are destructive, blasts are the precursors, the formers. Right? So this cell, the precursor to all the formed elements, all the formed blood cells. So at this point, it has committed itself to becoming an erythrocyte. So it becomes the hemocytoblast, gets the right signal to become a pro-erythroblast. We go to the early erythroblast, late erythroblast, normal blast. There's something very significant here that I'm going to come back to. And then eventually we end up into our erythrocyte. So again, we cannot have erythrocytes without this process, and we cannot have this process without that initial committed blood stem cell that we call the hemocytoblast. So important cell. And you can envision situations like different types of cancers, of radiation damage to the bone marrow, where these cells become damaged. And if this cell becomes damaged, every cell along the track becomes damaged, and you cannot manufacture the correct red cells there. So important. All right, and that leads us again to our discussion on the erythrocytes. And again, I'm going to elaborate on some stuff here. Form fits function, right? We talk about this, right? And if you can envision like maybe a frisbee, right? A frisbee that I throw into the air, it has a certain shape that allows it to move in air, right? Well, this is not going through air, it's going through a liquid, but the shape is significant. It's smooth on the sides. It's not going to get tangled on anything. It's just going to move. Maybe it might tumble, but it's going to have low friction as it travels through circulation, right? These red blood cells or erythrocytes. And we say they are biconcave discs. If you ever walk through the meat department at Albertsons or somewhere, you see where little kids go in there and put their thumb into the hamburger meat. That's what it looks like, but on both sides. So it's a biconcave disc, biconcavity on the sides. This is what's strange. They have no organelles. Right? And this is where I want us to be a little bit analytical, right? So we have started with a huge organelle, a huge nucleus. In the process of maturation, that nucleus gets ejected. So in essence, we have a cell that has gotten rid of the nucleus. Like think of a body getting rid of the brain, getting rid of mitochondria, getting rid of, Golgi apparatus, endoplasmic reticulum. All that is gone from the cell. Right? So that's kind of strange, right? It does come at a price, right? And I want you to kind of consider, why would a cell give up its nucleus and other organelles? It's basically committing, not necessarily suicide, but it's causing a very, very shortened lifespan as the cell loses its ability to undergo mitosis, repair, in any of that situation. So erythrocytes tend to be very short-lived. They don't live very long because they have lost their organelles. Any ideas on why we would have to lose mitochondria? Yeah, I'm curious to see if you can, link 1306 stuff with anatomy 2 stuff. Why would a cell give up its ability to generate energy, right? Mitochondria generate ATP. Why would this erythrocyte not have that? If you answered, well, mitochondria require oxygen, right? Require oxygen to sustain themselves. The whole point of an erythrocyte is to transport oxygen. So it doesn't want to have any mechanism internally that's gonna diminish its ability to do so. So it's a very specialized type of cell, very good at what it does, but that came at a particular cost, a price to the lifespan of the cell, and its ability to repair. So basically, the cytoplasm is just hemoglobin. It's this liquid just loaded with hemoglobin, and we're going to discuss that in a bit. And there's the ranges for you nursing types. You can look at those. Hemoglobin. So hemoglobin, what is hemoglobin, right? Hemoglobin is a protein, right, involved in the transport of gases, both carbon dioxide and oxygen. Hemoglobin consists of 4 parts, actually. It's a 4 repetitive type of structures. So we have 4 globins, or we're going to call them 4 polypeptide chains. Very fancy way of saying 4 proteins. So 4 globins, 4 proteins, and embedded in each globin we have a heme group. So this is where we get the name. A heme teamed up with a globin, we call that then hemoglobin. So each hemoglobin contains an iron atom, and this is what makes them heavy, and this is what enables it to bind with oxygen as well. So again, the range for hemoglobin there. And so here we have the globin, this sort of tubular chain there, and then that little red heme in the middle, and in green they're showing Fe. This is iron, right? So we have the iron in the center, a heme group surrounded by the globin. So that's 1. Another globin with the heme and the iron, 2, globin, heme, iron, 3, globin, heme, iron, 4. So that's the repetitive nature of the hemoglobin, right? And again, we have a bunch, right? There's a bunch of these throughout. We're talking about millions of these hemoglobins within that cytoplasm there. So we have a neat little system of transport. Oxygen has a very high affinity for binding with iron. We have a lot of iron, assuming you're not anemic, right? And we have this repetitive nature within these erythrocytes. So let's go back and revisit the red blood cells. So we said red blood cells have a short lifespan. And well, how short? We're talking about 100 days to about 120, depending on how active the person is, how much exercise the person does, how much stress the person is under, how hard these little red blood cells are crashing through the blood vessel walls and that kind of stuff. Yeah? So They don't live long, and every day we are killing off about 100 billion erythrocytes, but that's okay because we're producing about 100 billion erythrocytes each day. So I don't know if you ever wake up just kind of tired, you slept well, but you still feel tired. Well, understand our body uses a lot of energy to constantly replenish these. So the red blood cells don't repair themselves. They have to be manufactured from zero, from scratch each time. Yep. So red blood cells are produced in response to the hormone, endocrine system again, right? So the integration of our endocrine system back in, endocrine system releasing the hormone erythropoietin, right? Erythropoietin is basically a very important hormone, right? It's regulated. It seems kind of strange, right? So we talk about blood. We thought, well, maybe the heart is going to regulate the amount of blood that it's kind of having to push, but that's not the case. Erythropoietin is actually triggered by the kidneys. So the kidneys are responsible for releasing this hormone, which tells the body we need more blood cells or we need less blood cells. And basically, erythropoietin will trigger the formation of more blood cells. So blood cell count, blood cell volume is going to be regulated very heavily by the kidneys, right? And by the corresponding endocrine related hormone erythropoietin, or EPO. Sometimes we see it called EPO. EPO is a sort of like an artificial derivative mimicked from the actual erythropoietin. So erythropoietin, let's put ourselves in a couple of different situations, right? So let's say that a person is, I don't know, we're right now in the COVID situation. So a person is struggling with the COVID virus, right? So it's affecting their ability to transport oxygen through the body. Well, the kidneys will respond. The kidneys will say, hey, oxygen content is a little bit low. Let's trigger more red blood cells, right? Let's trigger more blood cells to transport oxygen more effectively. And that, again, that's linked to that hormone erythropoietin. So EPO produced in response to hypoxia. Hypoxia defined as a low oxygen carrying capacity of the blood. So for whatever reason, the body is not transporting oxygen, enough oxygen. The body, the brain senses a little bit of a deficiency. The kidneys then get the signal to start releasing the EPO. Bone marrow starts to manufacture more hemocytoblasts, go through the process to make more red blood cells. So it's this this feedback mechanism that you should remember from 2402. So hypoxia. Well, what can cause hypoxia? We have just an absolute low number of red blood cells. Let's say I had a bad laceration yesterday, right? Nasty cut, lost a lot of blood. Well, my body's going to be able to replenish that blood. Maybe you went and donated blood. Maybe for ladies, maybe during the menstrual process, you've lost a lot of blood. All that has to then be replenished in the body. So by the process of this hormone-derived EPO message from the kidneys to the bone marrow for the production of blood cells. Insufficient hemoglobin. Well, maybe you're deficient in protein. Maybe your diet is not real good. Maybe you're deficient in iron, right? You're anemic. So all of these can trigger low oxygen content, which will then stimulate EPO release to kind of get that back up to normal. Now, maybe you're perfectly fine, but we go up to Colorado, we go to Cloudcroft, to Rio Doso, and we go up into higher elevation. In a higher elevation, the amount of oxygen in the atmosphere is, or I should say the pressure of oxygen in the atmosphere is less. So our body will respond, hey, there's not as much oxygen. So being in high altitude areas, we start then to produce more and more and more red blood cells. Right? So it's a natural process due to people that live in high altitudes, training in high altitudes. So if I'm going to train to run the marathon in the Olympics, I'm not going to go to Houston, which is close to sea level. I'm probably going to go to Colorado, Denver, right, which is much higher up in altitude. So you're doing the same amount of mileage, same amount of workout, same effort, but our body's going to respond by making much more erythrocyte content. And the more erythrocytes we have, theoretically, the more oxygen they can send to these hard-working muscles there. So it's a very, very common trick in the realm of sports and athletic training and that kind of stuff. So, all right, so let's revisit some of these sort of hypoxic situations, right? What can cause hypoxia? We said all of these can, right? And all of these can trigger EPO, but specifically, there can be some disorders, right? There can be some flaws, some problems with the actual erythrocyte. So anemias are a collective term. There's different types of anemias. It's not just one thing. So anemia is a condition in which the blood has a lower than normal oxygen carrying capacity. So for whatever reason, the red blood cells can't carry the normal amount. So let's say, I don't know, normally if I go to the gym, I can lift a certain amount of weight, or I can squat a certain amount of weight. But maybe that day my ankle hurts, or maybe that day my arm is hurting. So I'm not going to be able to lift the same amount of weight that I normally do. Or say a truck can normally carry this many pounds, but maybe the truck has a flat tire that day. So it's not going to be able to function properly. And again, that's kind of how erythrocytes work. There could be a flaw with the actual hemoglobin, a flaw with the heme, any flaw with the actual cell, and that would be then considered an anemia. So low red blood cells. So this can be due to, again, bleeding, a bleeding event, hemorrhagic damage to the cell itself. There could be some genetic factors. There can be some radiation factors that cause trauma to these red blood cells. We mentioned there can be an iron deficiency. So different types of dietary anemias that can be present. And then there can be some genetic factors as well. So if, I don't know, we're in El Paso, but if you can link your genetic heritage back to the Greek islands, I don't know if I have any Greek students in this class, but if you can link it back all the way to Greek lineage, Greek heritage, possibly some of the Sardinian islands of Italy, you might be at risk for some of these thalassemia type genetic disorders. So these are genetic cellular disorders of the red blood cells. African Americans are likely to, not likely, but as there's a predisposition, there's a genetic factor that links them to sickle cell anemias. So these you would have no control over, right? You would just have bad luck, inherit the bad genetic instructions, and produce then irregular, improper red blood cells, which would leave you with a chronic state of anemias there, right? So the others are typically more acute, whereas this tends to be more chronic, right? You have it since birth, and it'll persist throughout the life of that individual. Anemias. All right, so erythrocyte disorders. So we know we're supposed to have this many, they're supposed to do this job. There's some ability to adapt in certain situations. But if there's a situation that happens where we have too many, right, we can have an excessive amount of red blood cells. We're going to call that polycythemia vera. Right? Polycythemia, an excessive amount of red blood cells. So if you remember that number, we should have a hematocrit packed cell count of about 45%. if the hematocrit is above 80%, you can imagine 80% red blood cells, little white cells, and not much plasma. So that means the blood would become very thick, almost very syrupy. And you can imagine how hard the heart must have to pump in order to get that thick, syrupy blood throughout the body. So again, that would put a tremendous stress on the heart. Blood pressure would be very high, and it could, again, trigger rupturing of capillaries and different things. So not a good situation. And many things in anatomy and physiology, we know we want to keep a homeostatic point. If it gets too little, it's a problem. If it gets too excessive, it's another problem. And that's sort of the idea of this polycythemia. So this is what the normal blood smear, normal hematocrit should look something like this, right? Red blood cells, we have a lot of space in there, about 45% red blood cells. Polycythemia, again, at about 80% hematocrit, not a lot of space, right? Not a lot of space for plasma, not a lot of space for these white blood cells. So it would also have a negative effect on your body's ability to fight off disease, that kind of stuff, right? Feeling very dehydrated as well. There are two ways to spell polycythemia. I've seen it two ways. I don't know what the official correct term is. When I see it as polycythemia vera, it's with an I, something to do with the, like in Spanish, the masculine, feminine, the O or the A kind of text. When it's by itself, you tend to normally see it with a Y. Again, maybe some of you might do some research on that. Let me know the official spelling, but we know what it means regardless. Polycythemia, an excessive amount of red blood cells in hematocrit, in circulation in the body. So what I want to do now, right? I don't want to jump into the white blood cells. Is going to make it a very, 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 very big file. So I'm going to stop it now. Stop the video now with red blood cells. I do want to upload a little sort of video dealing with polycythemia as basically purposefully induced polycythemia. This is very common in athletics and in sports. And I don't know if y'all remember Lance Armstrong. It's not, kind of a little bit old school, but the stuff that he did was quite amazing, pushing the boundaries of fairness, of rules, and also of health, right? The kind of stuff that he did. So I want to upload a video that shows some of that and link it to then this red blood cell lecture. And then I'll have another lecture that I'll get done later this evening that will be on the white blood cells. So it will be broken up into two parts. Well, more. It's such a big, so we'll do red blood cells, another discussion on white cells. It's a big one. And then we'll address platelets and kind of finish up with that. So I will get through everything before, it's due. I will be traveling this next couple of days. So when I have time, I'll sit there and get these things done. But you will have them available, in time to complete all the homework that will be due on Monday. So with that, let me stop this one. And I will start to upload this onto Blackboard as well as the Lance Armstrong videos as well. All right.